Hi, my name is Catherine Lape, and today is June 26th, and this is my Unit 3 presentation over the non-coding DNA mystery. So basically, the non-coding DNA mystery, it doesn't provide instructions for making proteins, but it turns out it's actually full of a lot of information and is no longer considered junk DNA. Or selfish DNA. So the genomic code explains the levels of non-coding DNA sequences and genomes, but the non-coding DNA that is found has to be viewed basically as vital information. They contain sequences that determines when and where the genes are turned on and off. They provide instructions for the formation of different kinds of RNA molecules. So there are three main explanations that were brought forward for the existence of non-coding DNA that was found in the article. So for one, Ono proposed non-coding DNA was junk. Doolittle, Sapienza, and Orgel and Crick suggested that it was selfish DNA. And because it mainly involved transpons that visualized as molecular parasites rather than having an adaptive function for their host. So this led to the understanding of overlapping transcription of long non-coding RNAs that originate from the majority of DNA sequences, and that plays an important role. So Bernardi's research mainly focused on how non-coding DNA is something more than just junk DNA and selfish DNA. Granted, it does not code for the proteins, but it shows that there is no problems for the transpons to be correlated with the host isochores to be active. So the whole journal article mainly followed and showed research on a variety of topics that are important for proving what non-coding DNA actually is and what it's associated with. So therefore, one of the first indications for the coding sequences are evenly distributed in the human genome, and it came from the observation that the majority of sequences initially tested were localized in the GC richest isochores of the H3 family. It established correlations between isochores and chromatin domains. It also was derived from the demonstrations that replicons located in an isochore family show either all early or all late replication timings. In that early replication, isochores are short and GC rich, and late replication isochores are long and GC poor. And second, the conclusion that chromatin domains are stable units of replication timing regulations, and that these findings indicated that isochores correspond to chromatin domains and also relates back to non-coding DNA. So this article does an amazing job at explaining what exactly non-coding DNA is and how it serves as a functional role in regulating gene expression through a formation of LADS, TADS, spatial compartments, isochores, short sequences, and genomic codes. It advances our knowledge of non-coding DNA by putting into perspective what this type of DNA actually is and how it is vital for us, and how it has been observed as something that is functional and as no longer as junk DNA, since it does not code for proteins. Thank you.